Hello. Welcome to the 2025 Stephen H. Davis Lecture. Okay. Two items of business first. We're having a reception immediately after the talk, Michael's talk, and everyone is welcome. It's over in Cohen Commons. And if you are a Zoom person, please send your questions by chat and we'll try to get to them. All right, this lecture is given annually by the ESAM department in memory of Steve Davis, who passed away in 2021. Steve was one of the founding members of our department. Interestingly, he was actually the first hire into the newly formed department, third faculty member, but first hire, okay, in the department. And his impact was significant over the years. Okay, his contributions to fluid mechanics and material science resulted in him obtaining membership in a number of societies and other awards. For example, he was a member of the NAE and NAS. Okay, he was also a good friend and colleague who is truly missed. Today, we are fortunate to have Michael Shelley, Professor Michael Shelley from the Flatiron Institute and the Courant Institute as our speaker. Okay, Michael is the director at the Flatiron, the director of the Center for Computational Biology. Okay, so I think with all those collaborators up there, we'll find out what he does there. Michael works on modeling and simulation of complex systems in physics and biology. His PhD is from the University of Arizona in applied math. Before going to NYU, he was a postdoc at Princeton and actually a member of the mathematics department at the University of Chicago. So he knows Chicago City a little bit. Okay. Michael's contributions have been recognized by numerous awards, and in particular, he is a fellow of the American Academy of Arts and Science and a member of the NAS. Okay, he will speak today on self-organization, flows, and transport in and of living cells. Please join me in welcoming Michael.
Thank you. Okay. So, as is customary and proper, I just thought I'd share a little story about Steve Davis. Many of you are younger, so you probably didn't know Steve, but Steve was kind of a fancy guy, really. And maybe about 15 years ago, my wife and I were staying at a very nice hotel in upstate New York. And we went into the bar to have a drink. And we were sitting next to this gentleman, and I kept looking over at him. And I said, finally, you look a lot like this guy named Steve Davis. And he smiled and looked at me and he said, and you look a lot like this guy named Mike Shelley. So he was just waiting for it. But we had a nice talk sitting there at the bar. And as a consequence, he invited me to come out and give a talk, which was about 14 years ago or so. I think which was the last time I visited here was because of the invitation of Steve. So anyway, okay, so what I'm going to tell you about is 2 or 3 of the problems that are elucidated by these wonderful movies. So these are all showing various types of dynamical phenomena that happen inside of cells, particularly, but not always, large cells such as embryos, so that's a worm cell. Inside of cells, you have mitotic spindles that are separating chromosomes. It's kind of an active structure formed out of polymers and motors. Of course, embryos develop into multicellular organisms. So actually, this is just nematode to nematode there as it goes through a whole bunch of cell divisions in the same volume that the single cell occupied at the beginning. Other types of embryos, such as in flies, houseflies, Drosophila, not houseflies, but fruit flies, Drosophila, they have these wonderful kind of fluid structure interaction problems associated with their development, with the development of egg cells for flies. Now, who makes an egg? It's the sperm and the egg together, or rather, who makes an embryo? It's the egg and the sperm together. And this from the same type of organism is showing you how fruit fly sperm is stored by the male in a little sac. And I'll talk about this at the end. It's a crazy little problem. And then we have other kind of beautiful problems. I call this the microscopic washing machine. So imaginative, it's small and it moves back and forth. But this is an artificial cell looking at how things that nucleate polymers get centered into the cell through mechanics. So it's kind of this class of problems, and plus the movies look great. Okay, but what's inside of a cell? I think, the old view was a cell was basically a bag of chemicals that underwent a bunch of reactions and somehow managed to duplicate themselves and form other bags of cells, of chemicals and doing things. But it's actually full of structures that help the cell move things from here to there, to position objects, what are called organelles inside the cells, very orderly and very kind of complex at the same time. And some of the main players are polymers. And so, for example, there's DNA, that's a very important polymer. There's also another polymer that's called, they're called microtubules. Its constituents are called tubulin, and it assembles into long, stiff polymers inside of cells. This transduces, these polymers allow the cell to transduce force from here to here because it wants to move something around. Another related related polymer is called actin. And this is just a kind of triple luminescence study, okay, of these different polymers inside of a cell. So some of this is what's called the cytoskeleton, is this whole set of ingredients that kind of handle all the mechanics inside of the cell. Polymers, varying degree of stiffnesses, cross-linkers, motors, these kinds of things. And they can self-organize themselves into structures that do jobs. A lot of what we're doing, we will be treating the inside of the cell. There's this kind of fluidic medium, it's called the cytoplasm. We're going to be treating this as kind of a Newtonian fluid and seeing how far we can run with it, through which objects are kind of being moved. But this is, if you actually go to electron microscopy, that's what the inside of a cell looks like. The cytoplasm, this kind of fluid interior of a cell, is like a slurry full of stuff, membranes, polymers, things like that. That's kind of kept fluidized by activity going on inside of it. Okay, but let me tell you about the first problem with that little preamble. So I showed you, I backed up a little bit, which I shouldn't, but I showed you this movie. And this is a movie of the cellular contents of what's called, it's an oocyte, it's an egg before fertilization, kind of moving around inside. Now, this is what it looks like at an earlier stage. That was a late stage egg. Eggs develop like anything else. There's a whole machinery inside of a fruit fly to develop eggs for fertilization. And there's a certain stage when that egg cell, this is inside of this kind of egg-making machinery, is called an egg chamber. When the contents start to spontaneously flow. Okay, so this is moving at about 100 to 300 nanometers per second. Doesn't sound very fast. This is not aerodynamics. The Reynolds number is around 10 to the minus 8. But one thing that this does is it cuts transport times of proteins or other types of things that need to get from one side to the other. It cuts transport times down to around 20 or 30 minutes. And otherwise, if we just relied on diffusion through this very viscous medium, it would take about a day. Okay, so this is a good thing, you would think. But how does it work? Why is it flowing? So it's a fluid structure problem. So does anyone want to say what's kind of weird about that movie as a fluid mechanics movie? Sir, what do you think? Yeah, you. Yeah, no idea. Okay, I do that just for fun. Yeah.
Is there not a boundary effect? Like, it looks like it's slipping along the boundary.
They're slipping along the boundary. There's nothing that looks like a no-slip condition here, right? So what's driving it must be going on along the boundary because it's slipping. It doesn't have a no-slip. If I had a rotor here in the middle and it was a regular fluid, then I would see like a big shear coming out from the boundary. You were a very good student. And so, so there was a picture put forward, and this is supported by a lot of experiments, including our collaborators here at Northwestern in the medical school, Wen Lu and Volodya Galfand downtown. But there's this kind of picture. Probably some of the details are wrong. Almost certainly some of the details are wrong, but it's an interesting model. So I told you about these stiff polymers, microtubules. It's known that they are sitting there bound into the side of the cell. And it's also known that if you shut down a certain kind of motor protein, a kinesin, so-called, these flows will stop. If those flows stop, the organism doesn't develop correctly after that. But here's kind of and this was a mechanical picture that was put forward from the Saxton lab, which we just kind of put on steroids, is that imagine I have a bunch of bound microtubules and I have little motor proteins, those are the legs. Motor proteins have legs, really, literally. So, and it's carrying a payload up this kind of flexible elastica. Then to carry that payload, there's a drag force on the payload, so there's a force being exerted onto the fluid. Newton's third law says there must be an equal and opposite force acting somewhere, and in this case, it's the microtubule upon which you're carrying the payload. And so you can imagine that there could be a compressive effect that causes something like a buckling in the system. And if it's somehow buckled collectively, so everybody kind of bent over in the same direction, you might incline the payloads relative to the surface and drive a flow. Okay. So what we did is we modeled that. And so that's what I do. I needn't have said that actually. But there's many ways in which you could model this. We're going to model the cytoplasm as just a Newtonian fluid, and it's filled with stuff that are doing things. And so there's the Stokes equations. How many people have taken fluid mechanics? Yeah, a good proportion. Okay, those are the Stokes equations, right? So we're going to have just a linear Stokes equation, but we're going to immerse within it objects that are moving through it. There's different methods you could use. I prefer kind of what's called non-local slender body theory for the Stokes equations plus boundary integral formulations for solving the Stokes equations. There's other methods. They're called it's called Stokesian dynamics actually, but there's other methods that are more Brownian. And then there's things like immersed boundary methods. So lots of choices you might have to make. But here's the basic model, okay? So we just take that picture literally. So what we're going to do is we're going to pin's the wrong word, clamp, language of elastica. We are going to clamp elastica onto the side of a wall. In the end, we're going to put it inside of a closed geometry. And we're going to have a model for motors carrying payloads and then exerting forces on these guys. And it's all going to be immersed in a fluid. And so this is kind of the discrete model. And so if I have the slender body theory for the Stokes equation, which is developed most nicely by Joe Keller and a guy named Rubinow some time ago. It looks something like this. This is a balance of forces. These are the drag forces of a thin elastica moving through a fluid relative to a background flow U, and I haven't said where that comes from. That creates a drag with an anisotropic drag matrix. And that is balanced by internal mechanical forces in the microtubule. Here, it's just an inextensible elastica. And so there's a rigidity attached to this term. And then this is the simplest model we could come up with, where we just say motors exert a force towards the base with some strength per unit, some force per unit length, sigma, along the tangent direction. So it's just a force pointing down the tangent, and that's it. So, and then, so this is the micro part. Each of these guys has that equation, that PDE. And then we sum up all the forces in the fluid that are a consequence of that collective behavior, and we solve the Stokes equations. And here, that's not quite how you do it. That's more of an abstract kind of representation, but that's what we do. And we solve the Stokes equations. That gives us the U bar. Okay, so that's the collective part. All these guys add together to create a flow that they all feel. Okay, so they communicate through the fluid. You can coarse grain that. You can turn that into a set of partial differential equations for an orientation field of elastica. And this has become a real workhorse in our group. And this is work I did with David Stein back in 2019. So imagine now that I just have this closed domain. Now it's starting to look more like an egg. And I just have a layer of polymers that are all bound to the side. And now I just have an extra index. Instead of I saying I have the ith polymer, I give a location along the boundary where that polymer is clamped. So arc length is an extensible. So this alpha and S are Lagrangian coordinates for this bed of microtubules. And here's the slender body theory. Again, now I've just appended an alpha to keep track of an index through this set. And then when you do the coarse graining to the fluid, you end up with, for this Lagrangian flow map of the polymer bed, this is again now the macro part where again the forces show up here on the right-hand side, but you're going from Lagrangian forces, Lagrangian frame forces, to Eulerian frame forces. And so you pick up the determinant of a Jacobian from transformation from x, y to this S alpha. With an indicator function, where are the polymers? With a density, how many are there? That's an areal density. And then you just go to town. What's nice about this is that, oh, okay, I'm going to skip this part. It has a nice energy budget equation, which I'll skip. But what's nice about this is that you can do analysis on these PDEs. So if I take a disk, then here's my polymer bed. It's really now a continuum. We're just sampling it here for representation. Then it has a exact solution where all the polymers are just pointing inwards along the normal. And there's a constant motor protein load across the whole thing. And so I create a negative tension just in my base state that's trying to, if I let it, the polymer bed would buckle. Okay. But if it buckles, the whole fluid flows, and then there's a coupling. And here's what happens. Just take a look at this. We're operating somewhere up in here in the polymer areal density phase plane and the motor strength. These guys, we put little perturbation on them in exactly how they're bent. Now, hopefully you can see they've all bent over. All right. And that movie is still running. I just want to say, I didn't stop the movie. So what's going on is that all these polymers bent over. When they're bending over, they push a big mass of fluid to go around. But once they're bent over, then all the motor protein loads are now on this canopy that you can see kind of running around, and it pushes a big flow. So this type of model, it's in its DNA, to coin a phrase, to create kind of cell-spanning flows. And there's more analysis that you need to do to actually say that as a technical statement, but we can. It creates kind of cell-spanning flows where the fastest growing modes are actually those that sit on the cell size. So now, at the time, we didn't know how to do that in 3D. So we did this, and this was with this group at Ray Goldstein and Eric Lauga, David and I worked with. And there was a PRL in 2021, something like that. But we didn't know how to do that in 3D. We didn't have the machinery for it yet. But we had all this machinery for looking at the interactions of discrete polymers in a fluid. Okay, it's something I've been working on since 2004. some of you probably, well, is anybody not yet born? Anyway, I've been working on it for a while. So what happens if you do this in 3D? And so what this is a cutaway of about 3,000 microtubules under this motor protein load model, sitting here in this fluid, immersed in a fluid, all pointing inwards in this spherical thing. We just cut it away so you can see what happens. Boom, there it goes. They buckle, you form defects that move around. These defects are actually the base of a big internal vortex, which is just spinning everything around. It just heads straight to the largest scale and forms a big rotating flow inside of that. And the form of the flow is that you have a sharp shearing boundary layer because just like you saw in that movie, everything happens at the boundary. So actually, if you looked at the flow profile, say on this left side, it's just showing streamlines, so you don't know it. There's a very sharp boundary layer, and then going up from the no slip condition, and then this is just like a big solid body rotation inside of it. You can't exactly do that in a closed geometry, so there are corrections, and it looks like solid body rotation plus a toroidal flow. Okay. So, and you can more or less, pretty much more, reproduce that whole phase diagram from this more complicated, kind of very computationally expensive simulation. This took about 3 million CPU hours actually to reproduce that whole thing. Well, it takes microseconds to solve the dispersion relation for the previous problem. But anyway, you do what you have to do. So you can reproduce that, and then you can play other games like ask, well, we did it in a sphere, right? Because it turns out in the sphere, you like to do things at very simple geometries at the beginning, the spherical cow, because then you can do other types of analyses in a sphere that you can't do in others. You can do like Landau expansions for simplified models, things like that, which we've done. But you might ask what happens in something that looks more anisotropic, like a real egg, okay? So this is a shape that we took from experiment. So it just looks like some sort of weird truncated bullet or something. And again, it's a cutaway. This is the top, this is the bottom, or you can call this one the bottom and this one the top. It's up to you. And you let it go. So it rapidly forms one of these vortices, which you see by those defects. Rapidly happens, and then it slowly starts to reorient over a long time. And it ends up forming a flow that goes like this. You can see it here. It goes like this. But it relaxed from a case where it already had a flow that looked like that. And so there's a 2 time scale problem, which I have not been able to identify the time scales, yes, but a good 2 time scale analysis of this has yet been beyond me. But it's this kind of beautiful problem of slow reorientation, accessing another time scale in the equations. Actually, it's the single microtube relaxation time rather than the bulk relaxation. But so we had this prediction that over long times, we should see reorientation of flows inside of these oocytes to being axisymmetric. We couldn't keep the stage 10, or I should say, we do some of our own experiments. Some of them are done at Northwestern, some at Flatiron, some at Princeton. And the Princeton side is done with Stosh Schwartzman's developmental dynamics group. So we couldn't keep the eggs, this is royal we, I don't do any experiments, but we couldn't keep them alive long enough. Once you excise them from the cell, you have to do that to do microscopy. You pull the whole egg chamber out to actually see that go on. So what we decided to do was to look at later stages in egg development. And so that led to very nice work by Alenka Jain, who's a Princeton graduate student that Stas and I co-advise. And what she did was not only do experiments, but she did experiments. This is a stage 11 cell. They start to look more and more ellipsoidal as they develop. This is visualizing what's called yolk. All heard about yolk in eggs. Anyway, it's in eggs. And took movies like this with various types of microscopy and reconstructed the internal flow field. And what she found was that in 23 of the embryos for this, 22 of them had axisymmetric flows inside. So it seems to be that as you go later in time, everything you see pretty much is axisymmetric, which, by the way, is a really crappy way to move things from left to right in a cell. It's kind of a mystery. So, but anyway, there you are. And she worked with a postdoc of mine at the time, Brado Chakrabarti, who's now at ICSD in Bangalore, on building simulations and theories for this. And it showed up in a PRX Life paper this year. Okay, that's the first story, little outline. Fluid mechanics, fluid structure interactions inside of cells. And I'm going to start that way on the next problem, and then I'm going to, at some point, dump fluid mechanics entirely and look at something else. Yeah.
I just asked, because you had up on your images this oscillatory regime also, is that
Yeah.
observed in cells at all?
That's a very interesting question. That's all I'm going to answer. So there are things about that experiment that you do see in cells, but so far as I know, not yet the oscillation. So, okay, let me take a moment and just describe that experiment. Okay, do you remember the washing machine one? So in a little microfluidic device, you have like a teacup full of what's called cell extract. You just grind up a bunch of cells and you get rid of all the big bits, and you just have this fluid. It's like the cytoplasm without the DNA and the membranes and all this. And you put in a piece of DNA and it will construct something. Okay, or you put in some other kind of protein and things will just kind of self-assemble out of it. It's really kind of amazing. So in that, there was a bead that was coated with a certain protein that causes microtubules to polymerize. So you throw this bead in there, it grows microtubules, they push on the cell boundary. And if the cell is wide enough, then it undergoes oscillation. If it's small enough, it goes unidirectional. So that unidirectional stuff happens, I believe, in something they're called monospindles, but you know, these are like little experiments where you get to see what would happen inside of a cell if you didn't regulate the way microtubules interacted with boundaries. Okay. Okay, so I want to go back to that first movie and read you my little, I call it a couplet, maybe? Okay, so just watch this movie. This is single cell C. elegans embryo. The male gamete has penetrated the cell, and what it's brought with it is DNA, which is now nicely encased in what's called a pronuclei. So that's the male nucleus. And it's got these two centrosomes, which are a little organelle, like maybe a centimeter, for God's sake, a micron or two in size, and it nucleates microtubules. And it's carried along with this male guy. That's the female. And I just want you to watch what happens. So the male starts off on the so-called polar side. They meet, male and female. They move into the center. The whole thing rotates while the mitotic spindle forms, which then elongates. And soon after this, you can start to see it here a little bit. The cell cleavage plane is starting to form, and it's going to scission that cell into two, leaving two duplicates of DNA on either side, along with a single centrosome. The next stage, those centrosomes will duplicate, and the same thing will happen again and again and again. Okay. Anyway, shall I read this? Say yes, please. Male and female pronuclei migrate, merge, center, rotate. Spindle forms, elongates, oscillates, chromosomes segregate. The cell then bifurcates. Anyway, I did that by chance, oddly enough, until I got to the last line. But okay, how's that happening? Well, as I said, coming in on this side of the cell is the male genetic material and a centrosome which quickly duplicates into two, okay, very early on. And it's shooting out microtubules. These microtubules, stiff polymers, their persistence length is well beyond the size of the cell. So they're they're stiff as far as the cell is concerned. And they hit the boundary, and either they're pushing on the boundary or something on the boundary is pulling on them or something. But this whole thing mediates, these centrosomes mediate the whole motion of this problem, of this dynamics. And what's actually happening, and this is the result of a lot of experiments, what's actually happening is that on the boundary of the cell, there are motor proteins called dynein. Okay, and dynein, there it is, there's the legs. Dynein are stuck up here on the boundary, and they'll grab onto an impinging microtubule, and they want to walk towards the other end. Now, they know which end is which, because microtubules are polar polymers. There's a plus end and a minus end. And these motor proteins have evolved to know which way is up. Okay, so they're trying to walk down towards this centrosome, and collectively, they're kind of pulling this whole thing along. And these microtubules that are being bound, that are kind of floating around in this whole thing are very transitory. They live for only 20 seconds. This whole process takes 5 to 10 minutes. So they're very transitory, which I think is good. It makes this whole mechanics kind of very flexible, adaptable. So let me just say, how do we know that the centrosomes are being pulled? So this is from a paper that we had last year. It was many, many years in the making. This is work done with Dan Needleman's lab at Harvard. Now Dan is part-time at Flatiron. And led first author by Haiyan Wu, who built a laser cutting device for microtubules. Very precisely can cut along two-dimensional planes and cut microtubules that might be connected to something like a centrosome. So if you can imagine, here I am on the side of a pool, right? And I have I'm drowning, so a bunch of lifeguards have thrown in ropes, and they're all kind of pulling me at the same time. I've grabbed every rope that I can grab, and somebody starts cutting those ropes, then I'm going to react, right? I'm going to get pulled towards the predominance of pulling. And that's what you're going to see here. So what you're seeing here in this picture is a schematic of a centrosome, and she's going to cut like a cup shape for all the microtubules that are surrounding it. The microtubules will rapidly grow back, but there's a period where there's no microtubules connecting it. There it goes. Look on the right-hand side. Cut. What happens is that centrosome goes flying towards the boundary, and then it comes back. The whole thing kind of heals itself. So that's telling you that there are pulling forces, and there are other ways in which you can decide those pulling forces are coming from motors and from dynein. So we built a whole technology around fluid structure interactions for different models by which this whole process, what's called pronuclear migration, could be happening. And it goes back to work in 2017 with Ehsan Nazikdas and Dennis Zorn and others, using a package that was called SkellySim, which really started off in 2004 with Anna Karen Tornberg. And so what we did was different models of how were motors being pulled, microtubules were pushing. There were motor proteins attaching to microtubules, like in that first model, and so on. And what we could do from that, and let me just show you a simulation. There's a pulling model. So there are motors on the boundary pulling on a bunch of ephemeral microtubules. And because of the geometry of the cell, it would get pulled into the middle, and then it would rotate into what's called proper position. So it did the correct thing. The problem is that every model did the correct thing. No big discriminatory power there. But what we could do is we could reconstruct the fluid flows that were associated with each of these different models, and they had different structures attached, different flow structures. So in this paper where we did all the laser cutting, Haiyan went and injected little nanodiamonds into the, this is just a wonderful subject, injected nanodiamonds into the gonads of parent worms. And then the progeny would have these nanodiamonds suspended in the eggs, and then you could use those as markers to do PIV, particle imaging velocimetry. So this is showing in a certain stage, this is showing the flow field that she measured in experiment. You may have noticed that when the spindle elongated, it oscillated. Okay, so this is that stage. So it's oscillating down, and you get this kind of flow that looks like just a big circulatory flow because you have this big object being pulled down. That looks very different from other models. And when we simulate that, we got a pretty damn good match on the structure of the flow fields, even down to having secondary vortices, kind of down at the end of the eggs. So, or embryos rather in this case. So we had this kind of picture of, because we did, there's other types of modeling we did, which I'll show you in a second, but large scale fluid mechanics, PIV inside of real eggs, laser cutting, things like that. Kind of said, yeah, there's pulling forces are the predominant force that is doing all this kind of work. So is that the whole thing? Okay. So I want to show you some new experiments. And this is done by Reza Farhadafar we have a little microscopy lab. And what he's doing now is he is tagging the centrosomes, these nucleating sites. You're not seeing all the microtubules, you're seeing the centrosomes, and he's tagged the nuclear envelope of the pronuclei. Okay, I just want you to watch this. So buried in here, there are two centrosomes that start off with one and quickly duplicate into two. There's two centrosomes. This is very typical. And it's just a segmentation here. Okay, it's not a model. It's a segmentation on the right. So there they go. Those two centrosomes move out of the cleft and come to the sides. They advance towards the front. And at some point, the female kind of rushes. They rush towards each other and meet. They squash up against each other. And then, I don't show it here, but they'll rotate. Actually, somehow that didn't show up there. I'll show it later. But the main thing here is that we treated the centrosomes in that previous work as being kind of bound to the pronucleus. We just didn't know what to do. But the main thing here that you see is that they move. So let's learn more about how they move. So this is more laser cutting. This is that stage where the two centrosomes are starting to cut forward. High N again. The master laser cutting is going to cut microtubules that are separating that centrosome. It's like a half a micron. It's just kind of amazing. And cut between that centrosome and the pronuclear surface. Just watch that cut. It goes flying away, and then it comes back. Okay. So it turns out that what's going on is that not only are there dyning motors on the boundary of the cell, all the nuclei are also coated with dyning. And so everybody is just every surface is kind of pulling on these centrosomes. Okay? And so there you go. And if now you do electron tomography, another kind of amazing technology. It's a little old-fashioned now, actually. But what you do is you will be looking through the light microscope. Fortunately, these things go slowly. You wait till a cell has reached a certain stage. You freeze it rapidly. You run to the other room. You dump it into a freezer. And then you replace all the water in the cell with plastic. And then you can burn things away with an electron beam and reconstruct the stuff inside. This is reconstructing the pronucleus and the two centrosomes and their microtubules. Okay? The main thing to notice about that, and this is from analysis, not from a picture, is that the centrosomes are mostly straight. They do not tend to wrap around the pronucleus. They're mostly straight. And so if they hit the nucleus, it's kind of line of sight. Okay? If they hit the cortex, or rather the boundary of the cell, it's line of sight. So what this sounds like is ray tracing. You have these kind of mobile emitters of light, if you want to call it that. They're microtubules. They shoot off in straight lines, and they hit motors if they're lucky. Okay? And they can't hit motors that are shadowed from them in terms of line of sight. That's ray tracing. There's shadows, there's things that are illuminated. There's regions which are doubly illuminated. So they compete for motors in regions where they're doubly illuminated. That's, this is me struggling for 2 hours with stupid ChatGPT, trying to get it to do 2 enterprises with tractor beams pulling on it. Well, it's supposed to be a board cube, but that didn't work out. So there you go. So we call that the tractor beam model. Here's the tractor beam model. Is that here are the centrosomes. There's motors on every surface. There's other laser cutting experiments you can do to show the female pronucleus is also covered with motors. That's why they rush towards each other at some point. And so you have shadowed regions. You know, this guy here can't access motors on this side. There's regions where neither of them can access motors. And this kind of what we call gating of the dynamics by basically ray tracing effects is very important to reproducing what you see in the experiments. Okay. Okay, so now this is where we dump all the fluid mechanics. All the fluid mechanics gets turned into a drag on the array of microtubules. Okay. So this is what we call the S model. S stands for stoichiometry. It's a biophysical fact that a motor can bind to only one microtubule at a time. So if I'm limited in my number of motors, that's an important effect. I don't get to just assume I can bind as many microtubules as I want to a motor. So all this is a drag term, which is balanced by the pulling forces of motors, each of which has a force m naught. There's m of them on the boundary. The boundary we call imaginatively gamma. We integrate over a density of motors. There's a probability that a motor is occupied by a microtubule. And the direction in which microtubules point is just the unit vector from the motor to the centrosome. That's the direction. And the force F gives you the force acted and just pulled on that straight line. Oh, it drives me crazy. I always try to make all the equations blue. It's my little thing. I dropped one. I find that very annoying. But okay, but that's kind of the picture. I have an evolving probability of motor binding on the surface, which I'll say about in just a second. I integrate over all those motor probabilities. That gives me a total force through the sum. That makes that guy move. Okay. So this P, it can be simple or it can be hard, but let's make it simple. What we calculate is, given where the centrosome is and how fast it's moving relative to the surface, we calculate the flux of microtubules from a Poissonian nucleation event to the surface. That gives me what we call the empty impingement rate to the surface onto a motor domain. Okay, and it's got a scale. Then at that point, in that little population of motors, kind of on that scale, there's a proportion that are available. They have not been occupied. The probability P is that it's occupied, and the proportion which are available in kind of what we call the independent motor model is just 1 minus P. Very simple. You can make it more complicated by assuming that the motors are distributed through kind of like throwing darts at a wall or something like that, or other kind of more complicated things. And then there's a rate at which they unbind. Okay, so that's very simple. But it works amazingly well. It predicts the scaling of spindle length. That model predicts the scaling of spindle length with cell size over 100 million years of nematode species separated evolutionarily. We used it to calculate the frequency of oscillation in agreement with experiment in that Nature Physics paper. We used it to look at the mechanics of human spindles in a DevCell paper last year. There's a theory paper we published in Physical Review Research, and we just submitted another paper on later stages of cellular division where cells getting smaller and smaller. So it's kind of a basic workhorse, kind of like with that aligned microtubule model. So now I said we used it to calculate spindle length. There's a spindle, there's the 2 microtubules. It turns out that A little bit earlier than this, you're in the stages called metaphase, where it's kind of sitting there at rest for a little while. And you can measure the distance between those two centrosomes. And then you can just ask the question, what's the rest position for two centrosomes inside of a cell in this S model? And it gives you a very nice proper scaling for it linearly with length, reasonable coefficients. But how do you adapt it? This is slightly interesting. How do you adapt it to have multiple centrosomes? So in this model, we're going to assume that if I have two centrosomes, like in real cells, not a single centrosome, then that population of motors, that 1 minus p part, how many motors are available when you're impinging microtubules from a centrosome onto it, is from the total population effect, right? It only sees what's available after everybody is had its lunch there, so to speak. So this is what the multi-centrosome model looks like. I have a position for each. I have a probability for each. And then in this equation where I ask what's the proportion available, I have to sum over the whole population to say what's available. That's the only way in this model they interact. And that's enough to do lots of things. As I said, there's two centrosomes in a theoretical model, 3 centrosomes. This is in this PRR paper. It's actually quite pretty. And why are they all kind of this? These are 3D, not 2D, okay? So this is formed some sort of triangle inside the cell. If I have 8 of them, it will form a twisted cube. Who's familiar with the Thomson problem? Oh, okay. Okay, what can I say? Education, that's just not what it used to be. I didn't know about it until a couple of years ago myself, but why they're kind of evenly distributed is because they compete for motors in finding their position. I just want to show you a little movie when we first started to work on this. We had much fancier models then, which chewed up far more time and gave far less light. But let me just run this. These are discrete microtubules in a fluid, and these are motors that they're bound to. And we start off 2 of them near each other. It's my creepiest movie, I think. They move away from each other. And why is that? That's because they both see the motors in between, and there's a finite number of them. While this guy sees all the motors out here, and this guy sees all the motors out there, so there's a force imbalance and they get pulled out. So they like to find a position where they have the least amount of competition for motors. And so there's a natural self-repulsion in the system from this effect. It all because they're competing for a boundary resource. Okay, so you put all of this into a model, the tractor beam S model, multiple centrosomes, multiple mobile bodies, all this kind of thing. You keep track of all the terminator sets that have to do with separating regions of illumination, double illumination. You build all that into a simulation. And this is what the simulation looks like. This is showing you the fields of peace. You can see there's a terminator curve here. One guy's illuminating up here, one guy's illuminating down here. There's a point at which neither of them illuminate. And it's doing all the things that you see, actually, in that first movie. And all we're doing is specifying a handful of parameters, 6, 7, something like that, many of them constrained by measurement, and just letting a simulation go. Okay. Let me play that again because I know you want to see it. There it goes. And the fact that these 2 guys move away from the cleft initially has to do with the fact is that they're competing for motors in between them, and they get pulled away in this cleft to opposite ends. Kind of explains a lot of things very nicely. And if you compare it with experiment, it does amazingly well. So this is the angle of separation between 2 centrosomes as a function of time in the simulation compared with data. That's the purple stuff in the background. Red is the theory, a single simulation. And you can do something that's called a GPR 1-2 knockdown, where you knock down binding of dynein to the cell boundary. And if we just adjust our density of motors, as we consider that a free parameter, then we can match that data. In the same way, using the same parameters again, we also match the distance between things. We match the rate of rotation. I'm not showing you the rate of rotation of the whole complex and so forth. And we've also just used it to explain spindle scaling dynamics inside of multicellular embryos, as you have to keep doing the same process again and again and again in volumes that are getting smaller and smaller and smaller. So you need to have a very robust way of doing things. And we adjust one parameter per stage, which is just the rate at which microtubules grow. Okay. Now I have, Mike, how long do I have?
9 minutes.
9 minutes. Okay, just right. Now we're going to switch gears. And I'll start off with this statement. Sperm are, I have to read it, sperm are among the most diverse and fastest evolving cell types. Okay, now there's the human sperm, the canonical sperm, because we're humans, studied to death in many, many different ways. But who would think that sperm come in a great variety of morphologies and operate in different kinds of environments? And so for the nematode, that's what the sperm looks like. This is what the sperm looks like for a fly, Drosophila bifurca. This is what the sperm looks like for something that I don't remember, but it's covered with thousands of flagella and it spins around. So there's this great variety of sperm, and there's interesting things to learn from. Okay, so I call these beyond the canon sperm. And there's a nice paper in Nature by this guy, Scott Pitnick, on why might you get kind of extreme forms of what he would call ornamentation, because males are competing for selection by females. So let's talk about the fruit fly. There's a male fruit fly, lovely, 3 millimeters more or less. That fruit fly produces sperm that are about 2 millimeters in length, almost its same size. And that's about 40 times the length of a human sperm. Okay? It stores those guys in kind of an order 100 micron sac, about 1,000 of them, into so-called seminal vesicle. And they need to be stored in a way that will make them, recoverable, because you have to use them to inseminate eggs. Okay, so they need to be stored in a functional state. So that's the question. How do you store 1,000 giant 2 millimeter long sperm inside of a 200 micron sac? Very simple question. That's what it looks like. That's minutes. What it shows is all the so here, there's just been a GFP labeling, fluorescent labeling of the tails of the sperm, 2 millimeter long tails. The heads are 15, 20 millimeters in length, something like that. You're not seeing those. But what it looks like is this big pneumatic mess, right? Everything's highly aligned and it's slowly roiling around. So my answer is you store it actively. So these are actually packed in here 40 to 60% volume fraction, which is huge. Fluid mechanics is not the game here. Those volume fractions they are called what's called highly screened. And so that you have this kind of slowly moving, deforming material, if I think of that as a material, has moving defects, and I missed the half going on here. There's a +1/2 defect that looks like that you saw at one point, and is propagating like a comet in the direction of its head. In the language of active pneumatics, this indicates something called an extensile stress in the system. Namely, there are stresses that are kind of stretching along the field lines of this pneumatic material. And this is work done by 3 people at Flatiron, and I already introduced Brato. We're missing an I there, Chakrabarti, who's now in Bangalore. Yasmin Imran, I see. I forgot to bring these to the front. Yasmin Imran Al-Sus did a lot of the observations, and then brought over the modeling and simulations by Bryce Palmer. Okay. So this is what the sperm look like when they're packed inside of the seminal vesicle. So here, this is something that's called serial block phase scanning electron microscopy. It's like a tomography thing. You freeze the cell and then you reconstruct the contents. And this is showing a section of several, we didn't do the whole thing, a section of these long sperm meandering their way, kind of you can see that these two are very next to each other and they stay so for, somewhere on the order of 50 microns or something like that, kind of wandering through this thing. And if you cut through, you can see regions, depending on the plane of the cut, where they're highly aligned, or if I cut this way, you'll see it's kind of packed with aligned sperm going through there. So it's a highly aligned structure. What happens if you pull one of these out? Cut open the seminal vesicle, the contents will spill out. This is a single sperm, and it has, well, the frame rate is wrong to see it, but it has traveling waves moving down it. But if you look at, they don't swim. They just kind of meander around. They're too long. They're kind of wrapped up. But it has these traveling waves that you see. So now, what I'm going to do is now, Yasmin labeled the heads separately from the tails. I showed you the tails. And there's the heads on the left and the heads and the tails together. And what you find is that the heads are moving about 5 times faster than the apparent material speeds, which you just take one of these defects and you see how fast it's moving. The heads are moving about 5 times faster. So this is a material whose microstructure is zipping around really fast relative to how fast the material moves. This is another ChatGPT creation. You're supposed to see that there are cars here moving in different directions. It took forever to figure out how to do this because it's not in its training set. It cannot imagine whatever that means. The cars would be in an arrangement like this. So I had a real hard time with it. But basically, individual sperms swim 5 times faster than the material moves, and they counter-propagate. It's like one lane after the next, somebody's moving in the opposite direction. Okay? And if you look at the histograms of material speeds versus head speed, you can see heads are fast, material is slow. So it's an active pneumatic material whose constituents are moving very rapidly, but they're in an ordered state. So let me just tell you that extensile stresses can disentangle entangled things. And this is from a paper of mine with David Santian and Alexander Zadovskov a few years ago, where we were modeling chromatin inside of the nucleus as an active polymer that had extensile force dipoles on it. So force dipoles that are pointing in opposite directions. And you start off with this as a simulation, obviously. And what happens from this extensile stress is that the material starts to order. So you went from this entangled polymer, you didn't lose the entanglement, it got kind of reduced into these small sets, but you see you start getting a lot of pneumatic ordering coming from extensile material stress. So let's look for it, and then I'll finish up in just a minute or two, I think. So the picture here is going back to Degennes and reptation tubes for polymer melts, is the picture that you imagine that each of the sperm is swimming in a tube made of all the sperm around it, some of which are moving in its direction and some moving in opposite direction. And it propels itself by pushing off the oppositely oriented sperm. And likewise, those push off of you. So it's a reciprocal arrangement. So there's a thrust, there's a friction. And in a model that balances thrust and friction in this kind of geometry, so here's the picture, there's reptation through a crowd, and this is quite different from pushing against the fluid. Here you're pushing against the crowd. Then you end up with this expression for the motion of the little material segment of such, which has a propulsive orientation q in a position x. There's material velocity, and then there is a velocity of motion against the background material that says, I need to look at my local polarity. How many people are pointing in the other direction that I can push against? And then that gives me my speed. So that makes a prediction. If I subtract this material velocity for oppositely aligned sperm, such, I project it onto the propulsive direction, then I should get that the speed at which those two will separate is independent of polarity and the material background velocity. And when you go into it, and Yasmin used laser bleaching to check that, and more or less, with some estimate of the polarity, is pretty much constant within huge error bars, which you could argue with. If you run with that model and you put it into a continuum model for a pneumatic do it in the micro macro part, so we do it in terms of a kinetic theory, then you can actually calculate what the active stress is from this counterpropagation. And it turns out it's an extensile stress, just as was indicated by the motion of these defects in the first instance and the fact that it was maintaining an ordered state. And via Daedalus, we did nice simulations of this model in a disk. We used Daedalus for all sorts of things, flows inside of spheres, models of the nucleus and so forth. Great tool. This is just showing what that dynamics looks like. And I think this is probably a good time to stop and just to say that this, we call it the active reptation model. It is in agreement with all sorts of details that have to do with the laser bleaching near defects and all these kinds of things. With the experiments, it's got a built-in separation of scales, so it's a little hard to argue that is a success. We built it in, et cetera, et cetera. But our conjecture is that these extensile stresses created by active reptation are what keeps this thing in a functional state. It doesn't let things become a tangled mess. You need this activity to keep it unentangled so that when you need them, you can kind of smoothly pull them out. And the same thing actually happens in the female. It's called the seminal receptacle, which stores sperm from various males. But again, they're highly packed and they look very, very similar to this. And thanks. For anybody in the crowd?
Hope that as long as you speak up, the microphones in the ceiling will catch you. So, questions?
Yes, please, Neelish.
So the cell division on the cell, if you apply some kind of a force field based on external field, would that change the orientation of cell division?
Yes, things like that have been done, actually. So there's a lab, and I don't remember the lab head's name, but they use laser heating to create thermal flows. And then they can modify where the spindle forms and its orientation by doing it that way. It's hard to use shearing on the boundary because these are pretty tough and tends to destroy cells, I suspect. Yes.
The idea of the flow within the cell is also contained in a fluid-like lipid bilayer, right? Does the flow itself?
I'm not understanding that first statement.
So is the lipid bilayer of the cell?
The cell membrane.
Right, it's fluid-like, right?
It has some compliance, but it's pretty tough in C. elegans embryos.
So they are treated like an anchored system.
Yeah, so that's right. I'm just treating it as a no-slip condition with no compliance. There's various places you could have compliance. You could have it into the cell membrane. There's also an actin layer that lines the inside, which it's immersed into. So there's compliance. In fact, that's an interesting thing that happens in other problems, that the membrane flowing actually lets motors flow into certain places. We don't model that here. We do model it in problems that have to do with nuclear envelope deformation. To reproduce certain experiments, you need to allow motors that are immersed on these pronuclei to move because they're being pulled on. Is that good?
Yep.
Okay. Yes, please.
For the last statement, it's a little bit counterintuitive as whether the extensile stresses are the things that help them functional, whether have you looked into frictional coefficient between those like biopolymers or anything like that?
Yeah, so this, I call it a conjecture. So this is the first paper that we've had on this. There's many things that one needs to measure. But one thing that we did do was to create 3D discrete element simulations, which I'm not showing you, actually, which simply have contact interactions without friction. So just contact, non-penetration, and you can get the separation out of that. If you have friction, you will have additional sources that should actually increase the speed. So yeah, I mean, on the modeling, we're trying to build more realistic kind of microscopic pictures. What you could measure from that experiment, I think we're mostly looking for consistency.
Because that kind of disentanglement question is also interesting for like textiles or like other kind of like multiscale
That's right.
systems. And we actually, together with Chris, we actually saw that the friction actually sometimes plays an effect, or like sometimes the shear locking angle would actually, like geometry would also.
Oh, that's interesting.
play
Okay.
a role, yeah.
I'd like to hear about that. All right, thanks. Did you have a question? No, okay, yeah.
All
Any more
good.
questions?
I had one. Kind of related to the previous question, do you see a potential explanation for like the elliptical shape of the egg as functional in terms of like aligning this mitotic spindle, or is that just?
Okay, so the egg is a very, thank you. So the answer is yes. But it turns out that if you take such an egg, so there are mutants in which you can get round eggs. And that thing aligns anyway. So it turns out there is yet another, I would call it a backup mechanism. And it's associated with some gene called LET99. And what that's associated with is actually in the kind of the coding of motors. I'm interpreting things now. On the inside of the cell, there's a band that is devoid of motors. And so you have a polarity breaking distribution of motors. And if you put that into the model, then it will indeed put things into the right orientation, even if it's a spherical cell. So there's a backup mechanism there. There's a whole interesting story about LET99, because it turns out that that kind of behavior gets inherited in the successive cell divisions by only one cell per stage. And it's the thing that becomes the progenitor to other types of cells, germ cells. Anyway, that's a long answer to a short question.
It's complicated.
It's complicated, yeah.
Other questions? And let's thank Michael again.
Thank you.
The day in which I need something to show.
因为我有 office hour 哦，对啊，我 I think